Where is this dude at? My goodness. My goodness. My goodness. My goodness. Lord have mercy. Mm, mm. What's going on, folks? What's going on? Yeah, I got wind of the Brain Trust the other night, trying to go over scientist Mel's video on evolution, and my goodness, I cannot tell you how painful. Like I did listen to about 10 minutes of it because someone gave me a call and said I listened to it. I started listening to it, and someone caught a particular interesting segment where G-Man's trying to correct scientist Mel's pronunciation of Neanderthal, and the fact that he has the audacity to try and correct somebody else's pronunciation of a topic he knows nothing about, I think it's hilarious. But no one tell him that she actually did say it right. And by the way, it's people. Some people say Neanderthal. Some people say Neanderthal. It is what it is. Uh, there is something very few people may not notice, but a lot of people say apoptosis for cell death. Actually, I've been corrected, and the guy who coined the phrase, so to speak, had a bit of a lecture. And this again, this comes from my instructor. Um, that it's actually apoptosis. Apoptosis. No, no, CRISPR. This is what's going on. Matter of fact, let me get you in here, CRISPR, because if you've been listening to Neffy and you're still awake, man, you deserve a prize. I have not listened to Neffy, and I think I did the other night, but he was talking about nothing. So I can listen to him try and whistle through his teeth and talk about proteins, and it's like, well, some of us actually work with proteins and some of us actually work in a lab, and we're like, eh, eh. Like I used to get. Let me tell you guys something. Let me tell you guys something. I used to get animated when I would come on here and I get a chance to talk to Brett Keane. If that doesn't date me, that I don't know what will. I used to get animated, come on here talking to Brett Keane and talking to like Neffy back when he was in his heyday. And there was a few other folks like me, guys like me, and like Star Splitter. For those of you who remember Star Splitter, we'd come on here and try and duke it out with them. But like once you actually have your hands in the shit, and you've actually worked like in a lab in any capacity, it makes all this shit funny. Like it really does, because you guys have seen the stuff that I do. And actually, if you guys have questions about some of the stuff that I do in the lab, feel free. Feel free. These guys get drunk as he is. Yeah. Yeah. So it's just it's funny because a lot of the stuff that they fuck up, it's not even like it's a part of a conversation that happens in the lab uh, that we laugh at. Like this is there's no serious conversation that happens in any major laboratory, even in academic laboratory. It's just like even in someone's garage cooking fucking meth. Like these conversations aren't happening. This little weird area of the internet is the only place that you'll find a guy who stacks barrels for a living and a guy who has a wig with the equivalent of a BP oil spill attached to it, talking about how much they know about a particular field of study that neither of them have any education on, formal or otherwise, and listen to them like denigrate said people in the field who, again, who've actually acquired some letters to some degree, literally. And it kind of makes you wonder, are they fucking stupid? Like, like I could curse at my wall for 9 hours straight and call Neil deGrasse Tyson every word under the sun, tell him he doesn't know anything about physics. He's just a big old poopy head just because he got taught by Carl Sagan. He don't mean shit. And like, who the fuck am I? Like, let's be real. Let's be absolutely. Can you turn on the air? It's hot as a bitch in here. Turn on the air. Like, who the fuck care? Like, it just let's be real. let's be absolutely fucking real. I had a lecture today on what? Let me see. Let's see if I was paying attention, folks. I had a lecture today on sterilization and using specialized techniques in a laboratory. Now, it doesn't sound exciting, and it's fucking not, but it's important. It's important to know how you actually need to sterilize your implements. You know, make sure that your media too, the stuff that you're working with, is sterile. Like, it's not so exciting stuff that's going to happen, but there is an important lecture because, well, my dumbass would have poured a fairly caustic substance into a filter, and it would have degraded the filter, and which we were cautioned not to because our instructor did it when he was a Padawan learner. So he doesn't want us to make mistakes like that. But it's like this today was one of those days where it wasn't too exciting. It was you know, how do you prevent contamination, and that is something that's not as sexy and it's not as appealing. You're not going to see too many scientist mail videos on sterilization. You're not going to see us having too many conversations about it. Probably not from Sam because he don't work in a real lab, but that's another case. That's another story. So for those of us who are wet lab guys and gals, we have to concern ourselves with really what are we trying to get after? I'll give you an example. Um, um 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 the Wonder Twins. Thank you, Sam, for that fucking. Who was it? Was it Sam or was it Bobby that gave me that? One of you fuckers called them the Wonder Twins, and I'm going to stick with that. So the Wonder Twins here, uh, they're offering their personal opinions, and I love this. I love this. I love this. So when you listen to like True Empiricism talk about specifically science, he doesn't do this too much with philosophy because he can't spell it. But when you listen to him talk about like what he thinks is happening in this field, I love the way that he like inserts himself into the field as if he has like a background enough. Based on my observations, yeah, you know, I think it's this. It's like, bitch, you can't spell observation. What the fuck have you been observing from your shithole? Like, I am dead fucking serious. Every time this dude says something like that, I want to piss myself laughing. I really want to piss myself laughing because he puffs his fucking chest out like he's actually done something in life. Like he's actually like look, some of y'all may not know what Science's Mill actually does in real life. That's just fucking complicated, all right? She explained that shit to me, and I'm like, you know what? I get what you're doing, but I don't get what you're doing. Like that's me being fucking humble. Like that's absolute. Like I could barely articulate what her job is, what her actual job function is on a day-to-day basis, which she gets fucking paid for, right? But I've been sent G-Man a link. I always send him a link, by the way. I send all these bitches a link, but they never come in. I went, Jesus is Lord. If you want to come in and just have a casual chat about evolution, that's fine too. Matter of fact, let me invite Spoken because Spoken was asking me. Actually, it was a pretty good question. It has something to do with uh, but believe it or not, let me go on a tangent. Spoken had asked a really good question. I'm not sure if it was genuine, but it was a good question about the rise of endothermy in animals, and particularly Animalia. You know, where did endothermy come from? And it seems silly, but it's actually a really good question. It's actually a really good question, and that's something that I go to. I don't know what is this website I have over here on my other screen that doesn't have porn? Oh, ncbi.nlm.nih.gov. Man, look at that. I go there and I look for it. Man, it's crazy. You fail to block another troll. Hey, they come up like cockroaches, Mel. They come up like cockroaches. It's okay. It's okay. Some of these guys are relentless. But what I do love is that while those of us who are actually taking the time, and I'm going to get away from some of the theatrics for a second. Some of us are actually trying to learn this in earnest, and it is a lot of shit. It is legitimately, evolution makes up a fraction of a percent of what like some of us actually need to do or need to know as far as our job or our perspective job. But let me give you a perfect example. I pulled this up. It's basically an analysis of the ribosomes for human PUM1 and its conservation. It's basically a histone that is this a histone? Let me make sure I pulled up the right thing here. Let me make sure. Because I was looking for histone conservation across different species. No, I think this is something else. No, this is for it's a RNA binding family. Yeah, I was pulling up different shit, folks. That's what happens when you see cool shit and go, you know what? That looks interesting. Anyway, the issue here is that you do have a fraction of people who genuinely don't understand what people have referred to as macro evolution. And as I see the idiots live, I'm ignoring them. There are people who genuinely don't understand, and they're like, okay, well, how, why we still got monkeys? That's the classic claim, why we still got monkeys. And some of them genuinely don't understand how different populations diverge, and that you will see a phenotypical change. But what gets left out of this conversation oftentimes, and even when I was tutoring, I really didn't see this from people who were trying to understand this. Again, people who are actually trying to understand this can have this explained to them. Every single time you hear these guys talk about this stuff, and Scientist Mel, you can use this. Please use this example. Please use this. You'll notice that you can tell when they're being dishonest. And you say, well, Drag, how do you know when someone's being dishonest? And, as opposed to just being mistaken. When you explain how you can have something categorized in many categories, but its difference lets it be like it'll let it be separate from everything else. It'll stand out because of that 1 degree of separation. You can explain how cladistics and shit works like that to people, and people get that. I am a male, I am an earthling, I am that's how many male earthlings do you know? How many male earthlings is it? Okay, I'm a father. Okay, how many fathers do you know? I'm a student. How many students do you know? Okay, all right, so we start getting specific. What sets me apart from everything else? Because again, I'm still with everything else. The same thing with cladistics. All right, the same type of categorization. So you can split off, let's say, well, compared to my wife, well, she's a female. She's also earthling. Okay, that's a whole nother category now. There's a whole nother category. So when we start getting to the end, when his example of, well, it's still a canine. Okay. When you call something a canine, you're referring to its genus. Okay. And it's for your common dog, Canis familiaris. All right, that's our woof. Okay, our woof. What we did. All right, those did not exist always. No, there used to be, was it Canis lupus? I think. Someone feel free to correct me. I want to say it was Canis lupus is the progenitor of Canis familiaris. Now, the breeds that we have aren't actually different species. They're actually just subspecies of the same Canis familiaris. I can't fucking say it tonight. Oh God. Anyway. Then there's a reason why. Dogs are Canis lupus familiaris. Okay, there we go. Okay. Thank you for that correction. When you start to diverge further and further away, you will take on different traits. So wolves are Canis lupus, dogs are Canis lupus familiaris. Okay. So notice and it's even in the way that we categorize it. They're still canines. They're still a part of Canis. They didn't leave Canis. Okay. But the subspecies is different. You have our dogs that are familiaris, and I'm going to familiaris, and you have lupus, which are still the wolves. That's why we still got monkeys. We still got wolves. It's not that hard. It's really Really not that fucking hard. Really not that fucking hard. But here is where things go awry, and I'll tell you exactly where it is. And as long as I've been on here, I can't believe it. It really took me a while to figure this out. If you go back further enough, like just if you go back to the furthest that you can go back, there's a point where mammals didn't exist. You're going way far back where chordates didn't exist. You know, backbones weren't a thing. You keep going as further back as you can fucking go, and these things didn't exist. Well, Drag, how do you know that? We got fossils. We can date the area pretty fucking reliably. The best techniques, the same, mind you, the same techniques, the same science that's used to put radioactive material in people's bodies to help cure them of diseases, to help detect certain diseases. That same type of science is used for this, by the way. Word to the wise. But anyway, they have an issue where you have to connect the dots, because you don't have DNA from 50 million year old fossils. That's not a thing. It falls apart. It falls apart. Just goes poof. All right, evaporates. They're uncomfortable making that connection. It comes from not because they cannot conceptualize it, because everyone can conceptualize, and I'll explain that in a minute. Because they have a religious issue. And G-Man and his little girlfriend True both gave it away at the very beginning. The first 10 minutes of their little chat. They have religious reasons why they don't believe. Now, every person who has taught me science has been a Christian. It's hard to believe. Only one of them would actually somewhat proselytize and inject her own Christian kind of creationist ID rhetoric in class. But she would couch it in such a way that this is just my personal opinion. She would actually acknowledge that this isn't science per se. This is how I see it. This is how I view it. And I respect her for that. I have no problem with her. I thought it was funny that she did that. I never made us think about it. But she would do it in a way that I found to be tasteful. We'll say that. So the issue with these guys, and the reason why they won't actually enter the realm of science, because they can't stay professional. They cannot stay professional. There's no way you're going to see BP oil spill wig in a lab. The one thing, he can't do math, as we see. That you kind of have to do. I'm sorry, folks. That was, and I'll give you an admission. Absolutely, full disclosure. One of the reasons why I did not actively pursue being a scientist right out of high school is because I thought I sucked at math. I don't. It takes me a while to get some of these concepts, but if I spend enough time with it, I'm going to learn it. That's just how I am. I really thought that I sucked at math, and I don't suck at math. I just hate remembering all these stupid fucking tricks that you got to learn. And someone knows what I'm referring to, someone specifically watching. But that's it. That's it. I'm taking my last math class that I'll have to take, and the only other math that I'll have to learn is the math that I need for my job. But you'll be doing it again and again and again. And I can tell you right now, I can tell you how to calculate how many cells are in any container. I can now do that mentally because I've had to do it so often. So it's I want to actually encourage folks, because I used to watch Bill Nye the Science Guy. I was that kid who stayed to watch Bill. I loved that shit. The sea monkeys. I loved it. I loved it. But that's what deterred me. And I said, I'm not going to be an idiot. I'm not going to let my personal fears deter me from what I really want to do in life. And that's what these idiots are doing. That's why they have to get on the internet and pretend. They have to get on the internet and pretend, oh, let me imbue myself with scientific knowledge. I'm sorry, Mel. You shouldn't have sat your ass in classrooms. You shouldn't have sat your ass in any labs for any period of time to take any type of certifications. You wasted your fucking time. If you were just on the internet making claims and pulling shit out of your ass, you'd be way more successful. Oh, wait. Holy fuck. Like, that's what kills me. None of these guys will actually ever take the time to do any of the legwork to actually get any type of credence behind their name, any type of actual knowledge. They will not. They're too lazy. They're too fucking stupid and inept to do it. And that's the fact. People can be upset with me saying it all day long, but that's a fact. That is a fact. If I had not lived that, I wouldn't be sitting here telling that to you right now. I sat in the fucking Mojave Desert with pre-calculus books with a chemistry book I bought off a fucking Amazon. I sat out there for 8 to 10 hours at a time. That's before I started going to school. Way before I started going to school. And it'd be another year before I'd actually get into any of these classes. So I don't like excuses. I don't like to hear, hey, Jesus is Lord. If you don't want me to cuss, that's fine. If you want to come in and have a talk about evolution, that's fine, bro. We can do that. I've invited you, actually. I have invited you, Jesus is Lord. Check your messages. I can't invite you on here for some reason because you're not coming up. But I sent you messages on mail.google.com. So someone just sent me a text message. G-Man just admitted to being gay. Oh, Lord. How are y'all getting my number? Anyway, so when I took the time, and I've learned, and this is the part that gets me. I was able to skate by, and when I mean skate by, I could have slept through all of my biology classes when I first earned my and actually, by the way, folks, I do have a degree. I just don't talk about it. I do actually have an associate's degree, but it's worthless, so there's no point in me mentioning it. But while I was working, trying to get because I was supposed to actually graduate with 2 degrees, but I couldn't take chemistry too, so that's why I only have 1 associate's degree. I was supposed to have an associate's in general studies and in biology. Well, that's what happens when you have a son. So I was also tutoring, and I've actually those are hours that I can now count towards things on my resume, and so I have built quite a rapport with people of letters, and text messages, and I can't actually explain stuff to you. That's not a problem if you genuinely want to learn. I learn from folks on here all the time. Jade taught me a lot. That's how I was able to skate through this. Jade, I learned so much from Jade. You wouldn't imagine. You would have no idea. I can't even count on all my fingers and toes how much stuff I learned from Jade. And because she actually spent the time learning this stuff, I picked up a lot of it. So, oh yeah, I remember Jade talking about that, especially with cladistics, especially with the way that we find the different ways that we can arrange the tree of life. Jade loved that sort of thing. So it's crazy. It's crazy how much of an impact that she had. And when I'm gonna say when I actually graduated, and as I mentioned to folks, I had learned a lot. I credited Fiona, Jade, Sam, Rational Mind, CRISPR, all of them for my success because I definitely did not do it alone. But it wasn't these idiots on here. Well, actually, except one, Tommy actually did help, believe it or not. The stuff that Tommy would say would actually appear on the first time I took genetics, and this is like population genetics. The stuff that he would say about genetics would appear on a test, and I knew it was wrong because Tommy said it. Hey, you don't have to believe that story if you don't want to, but I'm telling you, that is the fucking truth. That is the absolute truth. I took a final exam for the first time I took genetics. Tommy's answers, like what is epigenetics? Like the stupid shit Tommy would say would be the wrong answers. And I knew it because Tommy said it. And I say he is the best involuntary study buddy ever. Ever. So it is funny. It's funny how these guys like to, for one thing, I do have to mention, these guys get off talking to women some kind of way that you can tell that none of them have ever seen the inside of a woman's mind at any point in time. So let me point that out right here. But if they actually wanted to discuss the topic, you know what we have? You know what we were doing? We were doing journal clubs. In fact, what CRISPR was me, you, Sam, RSC. I don't think Doc Savage made that one. There was it Rational Mind was there, and we invited them, and we said, hey, let's just talk about. And we've done this several times, folks. And because it's like the evidence is there if they want to find it. The evidence is there. If they actually want to discuss science with us, we can do it in a way that we treat them better than they deserve. Because the way that let's say, I'll give you a comparison. The way that I discuss science with the TA that's in my class, there's a guy who he's a graduate student, and he's working at Baylor University doing some high-speed shit. The way that he and I talk is completely different than the shit that I hear here. There's not even a remote comparison. And this guy isn't, this guy who walks around with his chest out because he's working at a pretty famous medical facility here in Houston, and a pretty famous one here, a school that people wish they could go to, and they're trying to, murder their parents and get the insurance money to go just to be able to afford to go to. But it's like, it's crazy the difference in the conversation. And this goes beyond just having a shibboleth, sounding like you can actually, talk the talk. But these conversations don't happen. It's not, and it's there's not an onus upon other people. It's not my instructor's job to beg me to tell him what I don't know. It's his job to give the information and be available. That's about it. If I don't learn, guess whose problem that is? That's it. That's it. That is it. That's the problem with these guys. They think it's up to everyone else who has actually accomplished things to teach them. No, 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 no. Why is it someone else's job to teach Gary Moore, barrel stacker extraordinaire, the shit that he can go and read right on the internet? We tell him, G-Man, hey, just go to NIH. Don't even don't pass go. Don't collect $200. Go to NIH fucking website. Go. They got plenty of fucking articles. If they're behind a paywall, say something. I'll tell you. I'm the first motherfucker. I will tell you. I say, hey Sam, I'm looking at this. It's behind a paywall. I need that. It's okay. Give me a few minutes. I get a PDF in my fucking Facebook minutes later. CRISPR's done it. There's a lot of people here who have access to that shit, and they download the shit and they'll put it up there. They'll put it on Google Docs. So that's not an excuse. Not an excuse at all. So this nonsense that there's somehow like a gatekeeper that's stopping them from learning. I don't get it. Like, that's not fucking true. It's just lazy motherfuckers, inept and lazy motherfuckers who don't want to put in the legwork, who want to throw shit. I want to know how. Yes, I would love to know how to get past the paywalls. I would definitely love to. Tell me all your tricks, because I don't need that kind of strife in my life at all, ever. Because I've already been given one website today, CRISPR, adgene.com, I think it was called. That's what speaking of the TA, he gave us that. I think it's let me check. Adgene.org, ADDGENE.org. We need to use this website if we're going to take, was it Dr. Santiago's? Yeah, I think Dr. Santiago teaches bioinformatics, so we're going to need this website. So I'm adding this to my folder that definitely says science shit. So if there's definitely something that I can use, I'm going to use it. I don't give a shit, because it's only going to help me in the end. But these guys don't see that. I guess they think like having someone correct them or someone give them information is a mar against their image. Like, I don't know. Write article, DOI, find the current Sci-Hub domain, search the DOI. Let me copy and paste that. Thank you. Yeah. I'm putting that as a note to myself. Thank you, CRISPR. Fucking sneaky motherfucker. Fuck Pan. It's science. This all the science. All the science. Y'all want to know how I keep messages, like how I remember stuff? I don't have like a notepad because I'm just gonna forget it. I send messages to my Dragnot account. I'm not even joking. I actually go on Facebook and I message Dragnot on my real account, and I send him information. That's how I do it. What's going on, Miss Star? So, what is he? Oh, hold on for a second. The end is near. Completely right. You know what, CRISPR? I'm not gonna be publishing anything, so just saying, just saying. I just need, I may need that stuff for uh, research purposes. Gonna watch Netflix. Uh-oh. Yeah. Stupid. Yes, Science Mail. Go Netflix and chill. Definitely. And if you're near Houston, there's definitely some Netflix and chill around this area for you. But anyway. So bottom line is, folks, if there is something you genuinely don't understand about a field and science, there's a lot of us who do know quite a bit, quite a bit, quite a bit. If I can't answer something, CRISPR knows a shit ton more than me about cellular biology. So I'm trying to get to that level that he's at. And so behind the scenes, I guess let me explain to y'all how it works between some of us. So let me give you X videos' research too. Yes, it is. Yes, it is. Yes, it is. So I'm trying to think. No. Okay, I give you a perfect example. If I am trying to find, I'm just hypothetical scenario, very very real scenario though. Let's say that I am working uh, next semester doing some research that I have definitely hinted toward. Okay, that has to do with tissue culture. We don't really have anyone in the community that does tissue culture other than moi that I know of. Well, if I am trying to look for a certain expression of a particular set of gene or genes, whatever, uh, and I need help with, let's say I'm going to use some sequencing technology, I'm going to hit up Sam. We do have a sequencer. They don't know if it fucking works, but we're going to take care of that later. But we do have a sequencer, is what I'm told. Okay, so I got Sam for that. If I feel that my what I call my useful TE, my trypsin EDTA, is not the best, to degrade my extracellular matrix, the thing that keeps the cell stuck to the plate. If I want, let's say, you know what, I want to keep the trypsin, but the EDTA, I want a more efficient chelating compound. I can contact Doc Savage and say, Doc Savage, do you know of a better chelate that I could use? And so there we go. I got Doc Savage there. Let's say I'm doing something, I don't know, with let's say something with antibodies, something with that, something to do with antibodies. I might hit up Science's Mail, right? If I'm doing, if I'm trying to, I'm trying to think of something to get CRISPR in there. I'm trying to think, CRISPR, I forgot what your specific background is. I know shit. I remember everyone else, but CRISPR, I forgot what your specific fucking background is. God damn it. I want to throw you in there too. Anyway, you get my fucking point. Like that is how everything, because it becomes a project, right? Because everyone's like, "Ooh, I can help this. Ooh, why don't you do that?" And so in the background, the stuff that you guys don't see, cancer. Okay, so perfect. So if I'm working, let's say the cell line that I'm working with is a neoplastic cell line. CRISPR more than likely has worked with quite a few neoplastic cell lines. I'm going to take a wild guess, CRISPR, but you've worked with a few, and neoplasm is cancer, folks. So I'm going to guess you've worked with quite a few. So if I'm trying to find a fairly decent priced neoplastic cell line, that'll be, worthy enough for this research, well then CRISPR would be the person to go to. Let's say that I need to do some gene editing. CRISPR might know how to do that, So that's kind of how everyone jives together. That is how we all sort of lean on each other's understandings, and we move towards finding solutions. So yeah, decent price. I don't, CRISPR, I'm working with ATCC, so make of that what you will. That's what the school works with. I'm not familiar with any other repositories that we have access to, but ATCC is where we get our 293 cells from. So, you know, so is one of the best. Okay, yeah, that's where we got our 293s, and I think we're going to be getting some melanocytes and some other fibroblasts cells from them in 2-3 months, I think. Yeah, melanocytes and fibroblasts. But that's all I know for right now. I'm going to actually update you as we continue. But so CRISPR now kind of has an idea of what I'm doing, and so if I don't want to use, let's say I don't want to what I call useful TE, because the TE that we have here is useless. If I want to use my useful TE, then I'm going to have to actually understand quite a bit about biochemistry. Actually, one of the prerequisites for this class, which I have not taken, but I'm good like that. So I need to hit up Doc Savage. Oh, wait. Okay, Doc Savage. Well, Doc Savage suggests compound X. Well, I need to find out where I can get compound X. And if it's caustic to the cells, which by the way, the stuff that I use is caustic. I can just I can drown out I can not drown out. I can neutralize their effects on the cells by adding more media. That's how we do that. But it is, if I don't want to have to do that and waste more media, I might want to find a more useful chelate. So one that does its job, but the cell doesn't hate it, so to speak. So again, this is how stuff in the background happens. This is how we help one another out. If CRISPR is curious what's happening, at the academic level, maybe he's doing a presentation on how neoplasm, the sort of study of neoplasms is taught. Maybe RC was doing research too. I think he was research doc. So maybe RC wants to know how things are being taught at the undergrad level. Well, I'd forward him my syllabi and say, here you go, so he can then have a direct line of communication to the very thing that he's trying to study. So again, stuff like that happens. That's how moves are made in the background when folks aren't live. That's how things are actually spoken about, because it's not a issue of trying to pretend like I don't know. Like, I'm not going to sit here, I guess, and try and psychoanalyze these, even though I do that like 90% of the time. I can't imagine what these guys think goes on in the background. I don't know what they think goes on in labs, even though you clearly see what the fuck I do. According to G-Man, I mix Kool-Aid. I mix chemicals. That's evidently me saying that I'm going to split cells. I'm going to split. Matter of fact, how about this? Hey, CRISPR, I got a question. So if I have 95% confluency, let's say, I don't know, in a 10 centimeter fucking tissue culture glass, would it be better if I split them 1-8 or 1-6? How many people actually understand what the fuck I just said? I am actually asking him a legitimate question. That is an actual question. But how many people actually understand that? Because when I say that I need to split fucking cells, and this genius thinks that I'm taking like what am I taking, a scalpel? And yeah, 293s. Yes, CRISPR. Am I taking a scalpel and I'm looking underneath the microscope and I'm splitting, like I'm just like a fucking dissection of a cell, just spill out this what does True say? All the organelles. There we go. All the organelles. Just take all the organelles and just spill them out everywhere. Like, is that what happens? I don't know. Like, I've yet to be able to do that. If that's a pretty high speed fucking technology you got there. I got something to count cells. I have a I learned how to use a Muse analyzer finally, so I don't have to use a hemocytometer. But no, like it's crazy what these guys think happens, because then they want to pretend as though they have right to talk about what I do in a lab, CRISPR does in a lab, what Sam does. Well, Sam didn't have a real lab, but Sciences Mel does in a lab. Like, what the fuck do they actually know? Let's be real. What do they actually know? So who takes them seriously other than their friends? And I want to challenge folks to do this too. When you see these idiots talk about this shit, and and I guess they think that they're educating people. Ask some of their friends, what have you actually learned from G-Man and True and whatever science thing that they think that they're doing? What have you actually learned from them? Because I could tell you right now, how many of y'all now know what neoplasm is? How many of y'all know the name of the cell line that I'm using? How many of y'all know now the name of one of the major companies that sells that sells the cells? Yeah, that's funny to say. How many of y'all just learned a lot of that stuff? I'm just talking. This is just off the top of my head. How many of y'all understand what a chelate is? How many of y'all understand what EDTA trypsin is? Like, it's just so. If we're talking about who can actually put forth information, I'm sorry, guys, y'all are at a loss. Y'all don't have the information to put forth, and plus being inarticulate, and half of y'all are functionally illiterate. It kind of doesn't act in your favor. Kind of doesn't act in your favor. So like, I don't know what to say. If you don't know to go, if you want evidence, what do you think let's say, okay, well, I'll give you a perfect example. All right, here we go. I want to know. I want to know if my cell line 293 has been used in any type of cancer study, even though they're not an immortalized line. No, I don't think so. I want to know if they've been used in any type. I want to see where's the evidence that they've been used to study cancer. What do you fucking do? What do you do? Oh, here's what I do. Y'all want to see what I actually fucking do? Y'all actually want to see? Check this shit out. This is gonna be crazy. This is gonna blow your mind. Let me screen share. This is gonna be so crazy. By the way, Jesus is Lord. I sent you an invite. I don't know why you ain't in here. I go to Google. Check out that shit. Did y'all see that? It happens so fast. It happens so fast. NIH. Okay. What is this shit? Okay. So let me type in neoplasm cell 2. No, probably shouldn't have to type in 2 or cell, but we'll do that anyway. All right. So what do I have here? I got the growth of 293 cells in suspension culture. I'm not using suspension. HEK. Yep. It's really aneuploid and other and like other tumor lines. Or actually, are they? Are they? I should know this. I should know if they're immortalized or not. That's bad. I should know that. I don't think they are. No, see, and I knew it was going to do this. It's just latching on to numbers in here. So, yep, it's latching on to just random numbers. Fuck. Let me type in HEK 293. I am so bad at this. All right, cytotoxicity assays on HEK 293. Let's click on it anyway. Why not? Why the fuck not? Anti-alpha gal gene antibody mediated tumor cell killing. It's at the bottom line. Huh. Well, here we go. If if this is pertinent to what I'm looking for or not, this is the evidence here. By the way, when you they are immortal. Okay, so they are immortalized. There we go. I'm tripping. So oh yeah, because they've been treated with the adenovirus. That's right. They have been treated with the adenovirus. Okay. So I had it right the first time. So if this is pertinent to what I'm looking for or not, folks, this is the evidence here. When we talk about evidence, oh shit, here we go. When we talk about evidence, here we go. This is the shit here that folks are talking about when you say evidence. All right. This not that specifically, but you get my point. Like this shit here, this showing what was actually done, this is the evidence. All right. This lets you know work was done. People didn't just fabricate these. Don't put a space in your search. Okay. Thank you. And I appreciate these tips because I suck at this. Don't put a space. Okay, so just put everything together. Put everything together like this. Looks silly though. Actually, let me just invite CRISPR. Might be easier. Oop. Okay, I invited him. You led me wrong. You led me wrong. I don't need cell in there. Ah, okay. There we go. Oh, okay. So it's supposed to look like this. All right. Thank you. Yeah, and see, that's the type of thing that I like to know. Makes these searches go easier. Now look at that. I got more results too when I put it in like that. Look at that shit. Look at this. A lot of these probably aren't really pertinent to what I just fabricated, but still, you get the point.
Yeah, I didn't mean no spaces.
I'm just being silly, but yeah, like this is actually interesting. I just made up something just to show folks. Like if we're talking about like evidence, I don't know what these guys mean when they say evidence. Maybe like you've been listening to Neffy, so do you have an idea?
Oh, I had that muted a long time ago. I was just watching the side chat on his. See if anything interesting came up.
I doubt it. I don't think Neffy has new material. Or does he? Anyway, so yeah, we're doing cryopreservation tomorrow, and I'm pretty sure I'm going to record it. Should be interesting. Oh, I was going to ask you, do you guys use trypsin EDTA in your lab?
Yeah.
Okay, so that's pretty much just a standard to something that to break up that fucking ECM.
Yeah, I mean that's a pretty common thing.
Oh, okay, cool. Well, at least because I'm always curious to see, you know, what type of techniques that we learn that will get carried over, you know, until like to graduate school. Oh, you guys get to see my fatness, by the way. You get to see it. Ah, 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 okay. I had to make it awkward. There we go. Oh god. All right. So if when you I lost my train of thought. So what we normally do is when we're trying to maintain our cell line. Well, I should say our subcultures. What we usually do is, or actually, have you even seen my videos? I don't know why I'm explaining to you if you've seen it.
I probably missed most of those. Like if I don't know what the actual content is going to be, I tend not to watch those. But I like to see like the big arguments and stuff. So that's just it's more amusing to me.
Yeah, because I'm pretty sure the shit that I do in undergrad is just like Yeah, it's just not really that interesting. I do it because Jade would want to see it, and if she's smiling down from Catholic heaven, I'm pretty sure she enjoys it. So, but but yeah, we're just trying to maintain a subculture. Right now, recently we learned how to use the Muse Analyzer and basically how in general flow cytometers works, which is fascinating, by the way. Very fascinating piece of equipment. But what was I gonna say? Lost my train of thought. What recent discussions have you been looking at? Killing brain cells.
You know, there hasn't really been anything really good lately. I mean, just I don't know why. It's just sort of a lag in the community, maybe, but it's just I don't know. I'm sure we'll have something good here eventually.
Well, I didn't listen to it all. G-Man, didn't he say that him and True are going to start a regular series, doing science talks or something?
Well, I hope so. That'll be great.
Oh God,
I mean,
dude,
not educational, but great.
just fucking funny. Like when they did their science hangout last time, not this one, but the one where hydraulics came up. Like, I shit you not, I forgot everything else that happened in the hangout, but that.
Yeah, well, and actually, you know, T was in Neff's room the other day, and they were going over chromosome 2 again. It's just the same wrong stuff as he's always been doing. It's
I can only imagine. It's what I personally love about TE. Well, Neffy just kind of prattle on. He has his little kind of script, and he'll prattle on about proteins and the complexity of proteins, and he'll say complexity again, and he'll mumble about something, and he'll mention design. He'll say complexity again. It's the same sort of thing. But with True, he'll go, well, based on my observations, I don't see any of this stuff. I think they're just he has this air about him as if he actually knows the fuck he's talking about. And maybe that's just funny to me, but that's my favorite parts. What? I'm looking at the what is okay, people are sending me something. Oh, what the fuck is okay? Someone said G-Man said some really gay shit. Hold on. Are you ready for this? Let me. All right. What is this? I do everything in my power to avoid gay sex. He did not say that.
Let me think of another one. Homosexuality is a sin I definitely can't stand, and I hate it with a passion. Let me explain what I mean by that. I'm not into the idea of two guys getting together and having doing things with each other sexually and whatnot, and calling that a relationship or whatever. You know what I mean? That is something that I hate.
What's he doing with his hand right now?
That's a good
as far
question.
as me doing any of that kind of stuff and whatnot, I fill my mind with the word of God. I retain God in my thoughts on this particular topic. I remind myself that scientifically speaking, it is unnatural. And the Holy Spirit was constantly reminding me about God's word, and it keeps me away from that.
What? Why does he need to? What? It sounds like he has some type of attraction towards gayness or gay sex, and that he's saying that he's using like religion to make sure that he stays away from it. Or did I misunderstand what he was saying?
It's one of the things that I've always thought was really funny is, I mean, he's in the group that wants to call homosexuality a choice, right?
Yeah.
Like, not a choice for me. Is it a choice for you? I mean, I don't like
paper or plastic, dicks or vaginas. Like, I don't know. Like,
that's not like, it's not like in the realm of a choice. Like, but maybe it is for him. Like, maybe He wakes up every day and he makes that choice that, he's not going to suck any dicks today. He's like, I've made that choice, not going to do it.
I see that. That's like, I'm sorry, but sucking dick just doesn't come across my mind unless I'm getting mine sucked. But man, that we learned something else about good old G-Man. I am going to use the shit out of that. Oh, I just realized something. That's how he got his UTI. Oh man, maybe he had a slip up, and he let the dick demon penetrate his thoughts. Mm. 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 It makes so much. All the hostility towards women makes sense now. It makes sense. Maybe Ronnie was wearing lipstick, not for me. No. But for G-Man. Maybe it's part of their nightly ritual. Maybe they coo and whisper to each other at night.
Okay, I gotta point out that the amount of time you've already spent on this is probably not saying anything great about you either. So let's maybe move on.
Yeah. Oh God,
that's a lot of thought you just put into that.
It's like I like to overanalyze everything. That's my hobby. But holy shit. So yeah. So how was your day doing actual science, Christopher?
No, fairly good.
So I do have a question for you, because I'm being, I guess you could say, mentored also by the TA, that there's a transition that needs to happen from just going through the motions and performing these tasks and transitioning into an actual scientist where you're moving toward a goal. You're not just here going through the motions. So could you at least walk some of us through the day of an actual scientist?
Well, it actually tends to be a decent amount of variety in it, is part of it, right? Because, I mean, first, there is a lot more writing than you think. Like, however much writing you think there is, there's more than that. Because, like, it's not just prepping manuscripts for publication, right? But you also have grants, right? You have just it's never ending. New protocols. You have to go through all the committees, just everything. But then, there's, you have to know how to manage your time with different bench work, depending on exactly what you're doing. But just to get to the point that your TA was trying to make, the difference is when you're just going through the motions, you don't really need to know or care why you're doing what you're doing. You're just trying to get it done. And that's fine if you're a lab tech, right? But if you're a scientist, you need to know and care why you're doing this. You need to know how this particular experiment is going to mesh in with all of the other work that you're doing and that your lab is doing and that the scientific community as a whole is doing, right? And how it is this is going to contribute to some publication at some point, or if it doesn't directly contribute, how it's going to, inform your future experiments, right? Like this. So it's a lot of but there's also just a lot of writing and record keeping.
So that's why you spend a lot of your day listening to idiots, so that it makes the time go by faster.
Well, I listen to very little while I'm actually working. I'd rather listen to music. Because if you're listening to some idiots going on about something while you're doing your cell culture, then what happens if they say something stupid and aggravating, right? Like you're gonna be tempted to just get up and like start typing a message away. You can't do that. You have to finish your work. So music's a much better choice.
That is true. I caught myself yesterday in one of my live streams. I picked up a micropipetter instead of the pipette aid, and ended up putting in 400 microliters of media instead of 4 mils. And then I felt stupid after I realized what I did. But I've been listening to idiots. So yeah, no, you were absolutely right about that. You're absolutely right, because it is distracting. It is distracting. Yeah. So take note, folks. If you learn nothing from this hangout, don't listen to idiots and try and do actual science. Doesn't work out.
Yeah. Do the actual science, and then when you get home, listen to idiots. It's a great way to unwind.
I see that. I see that. I'm going to have to stock up on Kahlua and vodka and half then. I see that in my future. All right, folks. I think Idiot's waiting to go live so he can tell us, or he can regale us on his knowledge of what is he's going into cell biology now. I think he said. I think that's what he said. Yeah, he's gonna be talking to the Brain Trust about cells and something to do with them or whatever. Cuttings. There we go. I think. Okay. So, did you also hear him say that he was going to cut cells?
I just want to know what cells he's talking about. I mean, I don't know if you saw in the side chat, but you know there's a macroalgae that gets about 10 centimeters long.
What?
This Acetobularia, right? It's a single-celled organ, single nucleus. Important distinction. 10 centimeter long.
That big?
Yeah. On. Let me grab.
Really? If it's that big, yeah, you can cut it. But yeah.
But it's not just round either. It's got a particular shape to it.
What the fuck?
They're really neat.
Wow. Acetobularia. Yeah, genus of green algae. Holy shit. Yeah, I'd heard of was it Pelobacteria? I think they get pretty big, but I think this takes the cake.
Well, I mean, there are a lot of things that are single-celled, but they're multinucleated, right? So
yeah,
in a lot of ways, it doesn't really count because it's how far away is it from the nucleus that often really matters. But actually, with this thing, it's actually using distance from the nucleus to set up a gradient to actually get that morphology. So, and I guess that if you actually remove the nucleus and then damage it, it'll still repair itself correctly for a while, which is also really neat.
Ain't life amazing?
Yeah, this you can definitely cut. That's really easy. It's really easy to split these cells. You just pick one up, take a knife to it. I mean,
yeah, if it's that big, yeah, you definitely can. But yeah, I don't know how I would cut my 293 cells. If you know of, actually, I'm pretty sure there's a way to do it because we can put shit in the. Well, I was just thinking no, because a blastocyst is fucking huge compared to the 293 cells. So I'm pretty sure there is a way to cut it.
I mean, you can do microinjection. I mean, it's not cutting, but once you've got them sort of held in place, I mean, if you have the right instruments, you can do it to a single cell. But I don't know why you would bother.
No, I don't.
And Fiona was talking about she was doing cryosectioning on E. coli, which is but then it's when you cut it, then it's in a fixed substrate, so it's not like the stuff isn't going anywhere, right? And also you're cutting this big section. So you're bound to get one, or you're bound to get hundreds in each slice. But obviously, you'll be above many and below many too. So
it's fascinating. Going to get some videos on that. I just like watching stuff like that and having those techniques in my skill set. I'm not sure why I would need it, but fuck it, why not, right? Because we just went over. I don't know if I'm pretty sure you electroporation. Excuse me, and transfection. And there's another technique, basically to get DNA into the cells, and then to screen them for expression. There we go. That's what I'm looking for.
what kind of cells? The
293.
Yeah. I mean, sure, you can electroporate. That's so. I feel like that's somewhat old-fashioned at this point. I mean,
yeah.
Like it's just. I thought everybody just switched to all the other transfection techniques. I don't know.
Well, he's so here's what he's doing. So he said, "Well,
solid technique, especially if you're working with immortalized cells that are a little bit more resilient. There's nothing wrong with it. It's just like
it's old." Yeah.
Yeah, but I mean, it's convenient though, I guess, in a lot of other ways, because you can basically just get anything in there.
Yeah, he mentioned that there was a more common technique where I can't remember the name of the molecule, but you basically form a micelle around the DNA, and then the 293 or whatever cells you're working with absorb that micelle. I can't remember what it's called right now.
Yeah, there are a few different chemicals that'll do that, and then you have just good old lentiviral particles. Those work well too if you're talking about something commercial.
Yeah, I think it was calcium chloride. I think it's what it was. Yeah, I think it's calcium chloride gets coated. It's I'm trying to remember this. I should have wrote the shit down. Well, actually no, I did, but my book bag's not here. Anyway, so his idea is that like we're actually counting cells. I don't think I told you. We're actually counting cells using a hemocytometer. Like, when's the last time you actually used one of those?
I use one of those all the time.
Really? You don't use a flow cytometer?
Well, right. Like, I don't have one next to my, I don't have one like right next to me. So it's there's a shared equipment that, that'll run through those tubes really fast, but I have to walk over to it versus pipette a tiny drop onto the cytometer and look at it for 10 seconds. I mean, I
Sure.
can count that pretty fast.
Fair enough point. So that's, yeah.
One of the things that you'll notice about that when you're doing it is the if you look closely, those laser lines that are etched on there, you'd say much more narrow than the cell, right?
Oh yeah.
Okay. Now pick up that slide and hold it up to the light, and you'll find you can really easily see all those laser lines with the naked eye.
Yeah, I've actually played with it when I was trying to sterilize. I was like using 70% alcohol, and I would hold it up. I was like, oh, I can actually see this. Is it hit the light just right?
See each individual laser line with the naked eye, and each line is thinner than a cell is. So the human eye can visualize with the naked eye human cells, right?
Mm.
Apparently not, but it's just it doesn't work that way. But it's still interesting.
I thought I was able to see. Well, let me correct myself. I can see them when they're clumped up and I don't trypsinize them properly. I can't see them.
Well, yes,
but,
I can see a bunch of human cells too, because your camera's on. Like, and also I happen to have my hand up at the time, so there's a bunch of human cells.
I'm gonna see how many people actually got the joke, but no, they're too busy fuck with Jesus Lord. Yeah, yeah, everyone's fucking with him now. But yeah, I think it is fascinating. So I am glad that because basically his idea is to teach us like the low tech way of doing things, and then say, okay, well, you get to play with the fancy stuff later, but just in case your lab is using the old, not like the Baker laminar hoods, they're using like the older ones. I can't remember. It's something in Wiley, whatever the old company was, with the big ass switches on them and not the push buttons. He wants us to get used to old tech and then gradually, use the new tech.
Yeah, I mean, just as long as you've got, just get really comfortable with your aseptic technique, then the rest of it's pretty easy.
Yeah, I've gotten pretty good at it. I actually have a video of Mrs. Drag doing it too, and so I'm watching her to make sure that she doesn't, pass her hand over the open bottle of media, making sure that she's not, reaching outside the vent hood to mess with anything. And it just becomes second nature. And I was actually surprised how fast that I picked up on not touching things outside of the flow hood and going back and messing with my media.
Yeah, and you just gotta, you gotta get used to the idea of the, like, how am I going to organize all of the stuff that I need to do what I need to do efficiently without ever needing to move over anything in any inappropriate way, so that just everything's nice and efficient.
Yeah, I still have the issue. So it might have been you or someone called me out on. I push everything to the back, but then I ends up blocking the vents in the back. So
just push it to the back and then move it forward an inch.
Yeah, right? That's what I started doing.
Yeah, because it's reflex that you're just gonna to push it. It's really difficult to train yourself to like push everything almost all the way to the back, but
yeah, it's easy to push everything to the edge and say, all right, I need this space, but it's like, okay, well, let me pull it forward so that it's not blocking the flow of air. And so yeah, that's what I've tried to do. So my normal routine is walk in, turn on the water bath, make sure that it's at 37 Celsius, take out the media, take out my useful TE, put them in said water bath, and then go over to the hood that I'm going to be working on with the 70% ethanol alcohol, spray the inside of it and wipe it down a little bit and make sure that there's still some in there so that it can actually work and not just dry it completely. Turn on or close the hood, then turn on the UV lamp and then turn on the blower and leave it there for a few minutes so that it can sterilize. And then look over my protocol while I wait for my stuff to actually, no, I'm sorry. Look at my protocol, make sure I have all my materials, and my materials are either in the basket next to me or in the actual hood itself, so that they can go ahead and be sterilized as well, so I'm not getting up and getting stuff in the middle of the experiment.
Yeah, I mean, and you just have to be prepared for the fact that, when you're doing, as you add new techniques to the list, that, some of them you're going to have to slightly adjust things. So you don't want to have it so rigid that you can't like break up that pattern in any way, right? You have to have a little bit of flexibility so that when a new protocol calls for some new, like, you have to have several extra reagents or several extra bits of plastic sitting around, right? Like, you have to be able to fit that in without it completely messing with your workflow. But that's the easy stuff. It's the, like you said, getting into second nature where you put your hands, right? That's the difficult thing. If you can get that set to be second nature, then the rest of it's no big deal.
Yeah, and that's what I'm trying to get to, that standard, to that I guess that level, to whereas I know that if I need to calculate the total amount of cells, for instance, in a 15 ml tube, if I have 7 mLs of media, then I can take my hemocytometer, take like 20 microliters, and then count each of the four corners, divide that by 4, multiply that times 10 to the fourth, and then multiply that times the 7 mLs, and that should give me my, I believe, my total number of cells in that tube. And hold on, someone's tagging us. So it's, oh God, yeah. If y'all want to correct Jesus is Lord on math, good luck. I'm gonna tell you right now. Fiona says, imagine trying to do tissue culture on a trawler at sea during a storm. No, thank you. That's okay. I mean, I would do it just to see if I could, but it's nerve-wracking enough to have just like this. He's got us kind of scared to the point that, like, when my sleeve just bears a little bit of skin in between the glove, I'm so afraid of like passing that over my media bottle, because in my head, skin cells are dropping off. There's microorganisms on my skin cells that are dropping off, and they're going into my media. And then when I open my media tomorrow or go check it out, it's gonna be fucking cloudy, like instantly.
Well, actually, the worst thing is that if you get it into the bottle of media, right? Well, the bottle of media is going, isn't going to be in the incubator, right? So like, it's not going to be cloudy. You're just going to keep using it and wondering why all of your actual culture is cloudy.
Oh God, say that. Oh my God. So, and see that shit right there, that bothers me, because I make it a habit. So right now I have we call them NUNC tubes, NUNC tube for cryopreservation.
Yeah.
I have a NUNC tube already in the minus. I think no, actually he put it in the cryo freezer already. But I put it in the minus 20, and I have created 2 flasks of my sub. I think it's been, yeah, it's 4th generation subcultured, right? And I think I did like it was like almost like a 95% fucking confluency. Like it was, I'm not gonna lie, I didn't check it because I was too busy doing something else and I forgot about it. So it was like, it was 95+% confluency. So I split the fuck out of that. Like I did, like, think like 1 to 6, and I did 2 flasks, because I said, well, I'm about to run out of media. Before I switch my media, I want to make sure that I have enough cells for this generation, so that when I do this next switch, let me just do 1 flask to make sure that I didn't fuck up my media. Because if I did, then I'll still have 1 flask left over that I can make brand new media. I can put my PSG, my T, I put my PSG, yeah, my MEM media plus the PSG, and there's something else in there. I can't remember what else, but basically my new media, and just in case I fuck up. So I don't know, is that the right mindset to have going forward doing this sort of thing?
I'm guessing you're missing the FBS, right?
Yes, fetal bovine serum. Thank you. That's what it was.
Yeah. That's always a fun purchase, by the way. Like you don't have. Do you have any idea how much that stuff costs per bottle?
I have no idea. Like there's just bottles of it sitting in the fucking freezer, and I see it and it looks weird, and I just say, all right, let me just touch the one that says student. How much does it actually cost?
The less expensive is about $350 per 500 ml.
What?
Yeah. the better brands and like the higher quality is like $650, $700-$800 a bottle.
You got to be kidding me.
Nope, no, I am not.
Holy shit. Wow. That's a lot, because people have definitely been wasting media. that makes me feel so bad. But I mean it's fetal bovine serum, so I can imagine how it's
Oh yeah,
acquired.
and
Is
also the price fluctuates throughout the year
why the
because
cows protest?
fetal bovine serum, it follows meat production.
Oh, that makes sense. That makes sense,
Like
though.
if there's a drought, if there's a severe drought in the U.S., price goes way up.
So it's best to buy some when prices are great.
Oh no, yeah, that's what most people do because you can, if you want to get a lot, then you can. That is not to get a lot, but a number of bottles from the same lot. Um, and then just keep it at -80. It's fine for a while. So, and a lot of people do that. They'll get a few $1,000 worth. But
Well, how long does it keep within the because I like we put ours in the -20. So how long can you keep it in there and it still stay viable?
Depending on who your supplier is, they'll have different recommendations. I would say that certainly 6 months, 12 months at -20 should be just fine. Some people will say longer. I don't know how much you want to trust that, but that's but I mean at -80 though, right? Like if you've got the room at a -80, then like that's just it's fine there for a long time. But most people don't have the room there, so they keep most of it, you keep it at -20, and you try to buy enough that you buy the right amount for not just one group, but usually several groups, so you get a nice bulk discount. So, but
Yeah, there's yeah.
see, this is the kind of thing that you have to actually get into because like purchasing is a thing too. So
Yeah, that's actually what our instructor was telling us about, where you'll have certain salesmen that will come by, and that you have to, like, if you are good at finagling prices and getting things for good deals, you will then be in charge of writing grants, and you will be in charge of purchasing, and that will become your job if you are actually good at doing that.
he's not wrong.
Oh man, you speak from experience?
I've found myself in charge of more purchasing than I really care to. But it's amazing how much things cost, right? And like here's one of the things that really gets me, right? We get I don't know, a large number of the insulated foam shippers, right?
Mm-hmm.
In a given month, right? And people pay to get the round polyurethane ice buckets.
Mm-hmm.
Like I don't know if you've seen the ones I'm talking about, right? They're really common in labs for reasons I don't understand. They're round. They're usually a color, like green, like a dark primary color.
Yeah, I've seen them. I just thought that someone like I've never really paid attention to them because I think my wife is holding random shit up.
Yeah,
Sorry.
those things. Yeah, and those things are expensive too. They cost a lot more than you would think, and you get free things that do the exact same job. Like, why are you paying for that? Use a foam shipper. That's like I don't understand things like that.
I just assumed that someone went to Home Depot. Like I did. Like there's just so many things in the fucking lab that, at like something like that, I was like, okay, well I think they put the pasture pipe that like the cylindrical pasture pipe holder. Like that's where I see it. Like it just holds those for whatever reason. But I'm not sure if they were there before my instructor got there, or just somebody just said, well, fuck it, I really like these, so we're just gonna buy them. What do you? Okay, my wife's just being silly. I'm away from the laptop. She's just holding shit up.
Follow that link. Tell me what you think of that price on that.
Oh my god. You gotta be fucking kidding me.
Yeah, that is something that is worth about $1.
Why? 150 fucking bucks? Really?
It's like, oh, it's got a lid and you can put liquid nitrogen in it. Well, yeah, you can put liquid nitrogen in the foam shippers too. Like, not as many times probably, but who cares? Just get a new one.
For the price of one of those, you could get how many of those fucking foam things? Like, it's, oh my god,
infinite, because they're free. They send them to you with other stuff that you pay for.
Oh my goodness. I am learning a lot, sir. I am learning a lot. Oh my goodness. Because yeah, I'm gonna be working with my instructor next semester, doing some I think did we speak about it off air?
I think you told me a few things about it, but I probably don't have that many details.
Look in the side chat. Oh shit. I think he hit the wrong button. Oh my goodness. Blind using of shockingly expensive things in industrial industry is often common. I guess so, man. I guess so, but $150 for one container, it's not even that special. But this, okay. All right. You all right?
Yeah, I don't know what just happened there.
Okay. So if you look in the side, can you see the side chat? Oh, of course, you're putting shit in there.
I can see the like last couple things that you said, but
okay, you see the creating part?
Yeah, from there.
Okay, yeah, that's basically it. So yeah, that's why we're getting the melanocytes and the fibroblasts. So
it's gonna be told 3D culture.
Mm-hmm.
Yeah.
So this class is pretty much the prerequisite to that, and
yeah. Well, I mean, you need to have your aseptic technique down first.
Yeah.
Like no, you've never done any culture at all. That's fine. I'm sure you'll pick it right up. Here's all the expensive stuff. Have fun.
Yeah, though he had mentioned this in our molecular genetics course, and and so he said, well, the prerequisite to doing that type of research is this class. So I said, okay, well, I'll just take it during the summer. So like, okay, cool. And then you can just join me in the fall. I was like, really? That's it? It's just this one class? Okay, cool. All right. So, and yeah, so I'm going to be taking that. I've also I got an invitation to come work at well, my name is being put forth, I should say that, to go work at Baylor, and that was pretty fun. I do need to take what is it? It's a lab course, and it's called Lab for the Eukaryotic Gene, and it pretty much goes over. It's a biotechnology class where I think Dr. Santiago just runs the gamut of different type of molecular techniques that you can use for, like mammalian fucking cells. And so that's supposed to prepare people to work in industry. So yeah. Anyway, oh man, it's 10:30. I'm going to go ahead and let the idiot say whatever he's going to say. Thank you for this information, by the way. I am definitely very curious to compare what I'm learning to what actually happens in a lab.
Yeah, and actually, just to explain a little further the thing I was saying earlier about Sci-Hub, if you You know, if you find that DOI, and you just take all the stuff after the slashes, like doi.org, and then there's a slash, and you take that, right? And I've got an article up in a different tab, so I'll just use this as an example. So it's everything after the slash usually starts with a 10, and then you put that after the .tw of Sci-Hub. It's a direct link. It'll just go straight to the article. No search required.
That's crazy. I'm kind of curious how you found that out, but you know what? I guess that's better left unsaid. All right. Well, folks, thanks for watching. I did my rant about stupidity, but I would much rather talk about actual science because it's more interesting to me, you know. So, holy shit, and it works. Complex trait from the polygenic to the omnigenic.
Oh, and by the way, Tommy's back.
Come on. Really? Really? What is he saying now?
He's under the impression that because multiple genes or multiple loci can contribute to a trait, right? And that in fact some traits may exist as only the additive effect of many small contributions, right? That is, there's no single big component to the trait, single big contributor to the trait.
So
They're all just, they want to say omnigenic, like it's, so in other words, like it's not just that there's 5 or 6 things that are all having big contributions, but like maybe the biggest contribution is only 1%, right? Or something. Well, then therefore you can't have evolution for some reason. I was like, no, because that means that it's actually easier to get an additive effect. Because if you change any one of these, and there's apparently 100 of them or more, or however many there might be, you get a bit of a change. There you go. That's it. You're done.
So I'm a part of me is curious as to what article he misread to come to this conclusion.
The one that I just linked to.
That one.
Well,
okay.
actually, no. He misread a popular level write-up of the one that I just
okay. I figured. I was like, say, because I was like, he found this. I don't know about that, but okay.
The thing, that link is the one that he was looking at. It's his usual stuff.
Oh my god. Oh. Tommy. And it's funny too. Like this, I don't think Tommy deserves any quarter because my instructor came from humble beginnings, even working at McDonald's, like on the weekends as a shift manager, to being a postdoc at like what's it, fucking Stanford or some shit, some Ivy League college, whatever. And here he is now teaching, right? So Tommy's just lazy and inept. So I don't feel any sympathy for him at all, or any of his fucking cock swallowers. Jesus Lord, I mean, you can complain all you want. I sent you the link. I gave you the opportunity to come in. You would rather argue about the math that you obviously can't do in live chat, so that's on you. Maybe when you grow some nuts, you can come in next time. We'll actually talk, because like I said, some people actually have decent questions, like Spoken, his question about the emergence of endothermy, which is actually an interesting topic, but
that he's gotten an answer to.
Yeah.
20 different times, I'm sure.
Oh yeah.
Just keeps fucking that chicken. I mean,
hey, it's a warm crevice, all right?
Like honestly, I mean, I know I've answered that question to him. Like, it's not even complicated. I mean, it's complicated, but it's not hard to understand. It's just there's like a lot of steps involved, but it's a gradient. That's it.
Almost like it's almost like there's a design behind it.
It's like how do you have this happen? Well, first you start with, like you're not maintaining body heat, right? And then you start maintaining a little because that's convenient in some circumstances. And now you have a gradient, and you actually have examples of every position on that gradient. Well, not every position on the gradient that's continuous. There's an infinite number of positions, but you have a nice sampling of this gradient all the way from, like none all the way up to mammals, right? Or birds for that matter. Or, it's just, you have to be just trying to not understand something to be what
and that's the distinction I was trying to draw before, because I mean I've met people with genuine misunderstandings because they had the misfortune of having idiots as science teachers who purposely misled them because they have a religious agenda. I actually have a young friend who was in that position, and her science, put this in quotes, teacher should have been fucking fired because he sucks. But I think you have people like these cats who genuinely are describing Pokemon evolution. Like this, every time I hear G-tards say like, why doesn't a dog turn into a cat? Well, no one's fucking ever saying that. If a dog turns into a cat, we have a problem. We have a huge problem. So, I mean, if your Charmander's turning into a Squirtle, if your cat's turning into a dog, if your life is turning into shit, there's an issue.
I mean, firstly, like that's just that would just be extremely weird. But I don't even think that's what G-Man's saying. He's asking why doesn't a dog give birth to a cat?
You know what? Because what are those Russian nest eggs called? Damn it! I can never.
Russian nesting dolls.
Is that what they're called?
Oh no, the Fabergé eggs.
Is that what they are?
I don't know. I'm just guessing what you're trying to say.
And then Fabergé may be something different. It's just everything Russian is either vodka or something really fancy and breakable. I don't know. My mind works really weird when it comes to other countries. Anyway,
if you're thinking really fragile, then Fabergé egg is probably what you're going for.
That's probably what it was. Well, when I think of eggs, I associate fragile to it for for obvious reasons. But I don't know. That was a weird kind of word association that I just did. I had to kind of stop myself. That's me overanalyzing things again. Anyway. All right, folks. I'm going on. Okay? The Matryoshka. Okay, that whatever Sabella just put in the fucking chat. That thing. I'm not even going to try and pronounce that. It's late. I've not eaten very much today. Edgar eggs. Fuck you, Abad. Fuck you, Edgar. That's mean. That's terrible, but hilarious. Don't do naked push-ups, folks, and get a UTI. Don't be like G-Man or True. Do something with your life, please. Please do something with your life. If you can't, reach out, get help, please. Thank you, Christopher, for joining me and actually offering some very good information that I will more than likely take with me into the lab into the future. I'm not even exaggerating, folks. I actually will take this information with me because it is actually useful to me. So thank you. Make it push-ups. But yes. So any last words?
No, I think that closing it out on naked push-ups is just fine.
That'll work. That'll work. All right, folks. Y'all have a good night.